Hello, I'm Helena Todorov. I work at the Data Mining and Modeling for Biomedicine Lab at the VIB and the Ghent University, and today I will present TinGa, fast and flexible trajectory inference with growing neural gas. Let me tell you the story of a cell. This cell doesn't live alone, it has sister cells that might be expressing new genes that this first cell was not expressing. And this I represented here with this purple transcript. Now, the cell might have other sister cells that might start expressing more and more of this purple gene. They might even start expressing these blue genes or these red genes. But if I told you that the difference between these blue and these red cells are actually that they're representing two different types of melanoma, then this story would become interesting, right? Because if we were able to reorder the cells, if we were able to identify the genes that push the cells either into this blue branch or into this red branch, then we would have potential gene targets for gene therapy. Well, it is nowadays possible to reorder the cells in such a way, starting from a single cell dataset, in which you would have single cells on which you have some kind of information, such as the genes that they express, for instance. And inferring a trajectory starting from this single cell dataset is called, maybe unsurprisingly, trajectory inference. In the last years, trajectory inference has become very popular. It was first introduced in 2014 with two methods, Wanderlust and Monocle, which were able to model very simple trajectories, linear ones, such as the one that I have represented here. In the following years, many more methods have been published, which were able to model slightly more complex trajectories, such as the branching that I showed you just before. More recently, some methods were generated that are actually able to model very complex trajectories. Because in the last years, the methods, the technologies have evolved, and scientists have been able to generate data sets that were larger and larger and even mix different cell types. And so now we need methods that are able to model these different cell types in separate trajectories. But now a question raises, what if I came with a new data set? How would I analyze it? Which method should I use? The first answer could be, well, you have these complex methods, why don't you just use them? Because they should be able to also model the more simple trajectories. However, in a recent comparative study that was published by Salins, Canut et al. In 2019, one of the conclusions was that these complex methods are actually tending to really overestimate the complexity of the data. Here I have a small example for which we have a gold standard. So we know the trajectory, we know what it is, and we know that it should go from these yellow to these green to these purple cells. And this is the result that PAGA shows on it. You can see that it identifies a trajectory that is much more complex than the one that it should have returned. And so we see a kind of gap appearing here. We kind of lack a method that would be able to model simple trajectories, but also complex ones. And since we're dreaming there, we might as well ask for a method that would be scalable to large data sets. And these thoughts led us to designing TinGa. It's a method that relies on a growing graph that grows and is able to adapt to virtually any topology. In the rest of my presentation, I would like to go over TinGa in 3 different points. First, how it works. Second, how could we measure its performance? And third, how it actually performs on 250 datasets. But first, how does TinGa work? It relies on the growing neural gas algorithm, which was introduced by Bernd Fritzsche in 1994. And I will show you how it works, briefly show you the different steps on this dataset that I have represented here in the background, which consists of cells that form a circle here, and next to it, a kind of tree-like shape. The algorithm is first initialized with two nodes that are randomly placed and that are linked by an edge. Then, at every iteration, one cell from the dataset will be picked, for instance, this one, and the node that is closest to it will move towards it, such that after 170 iterations, you will end up with this. Your graph has moved, but you can also see that the size of the nodes has increased. And this is representative of the error associated with the nodes. Every time a cell is picked, the node that is closest to it will not only move towards it, but its error will also get updated by adding to it the distance between the cell and this node. Then, after a certain number of iterations, 200 to be precise, a new node will be allowed to appear between the two nodes that had the highest error. These nodes will again start to move, and you will see that new edges will appear between the two nodes that are closest to a cell. And more nodes will appear, they will move, and we'll see a new behavior in the graph. The edges are changing color. This is representative of the age of the edges. If an edge is not activated for too long, it will start aging, and once it reaches an age of more than 200 iterations, it will actually disappear. So all of these behaviors, the fact that the graph is allowed to move towards the data, to grow, and to get rid of things that it doesn't need, will allow it to eventually fit the data structure very nicely. Now we know how it works. A question that we had was, how do we measure its performance? Luckily for us, this comparative study that I already talked to you about also came up with some metrics to measure the performance of trajectory inference tools. The first of these metrics is correlation. What will happen here is that every cell of the gold standard will be mapped to this trajectory. So we end up with a kind of necklace that we can unroll. We'll do the same with the TinGa trajectory. So we again end up with this second necklace, and then we will compute the correlation between the cells. And as you can see that in this example, you had TinGa that found this sort of hook that shouldn't be here. Well, the beginning of this trajectory will slightly mismatch the gold standard, and so the correlation score will be slightly affected. The second metric, the HIM, takes into account the topology. What we have here is that in the gold standard, we have two nodes of degree 1, and between them, one node of degree 2. Now, what do we have in the TinGa trajectory? We have, again, two nodes of degree 1, and in between, a lot of nodes of degree 2. So these topologies are exactly the same, and so the HIM score, in this case, will be maximal. Then we have the F1 branches metric. This one will see if the cells were correctly assigned to the correct branch. In this example, it's not very accurate because we don't have any branching. We have two linear trajectories. And so in this case, all the cells would get assigned to the same branch in both cases, and the score would again be perfect. Finally, feature importance will look at the genes that are expressed along the trajectory, and we'll see if they are expressed in the same way in the two trajectories. In this case, on the right, some genes might be affected by, again, this little hook that we have here. And so this might, again, have a small influence on the feature importance parameter. Finally, with these four metrics, we can compute the geometric mean of these metrics and end up with a mean score, which is the score that we will use to assess whether a method resulted in a trajectory that is correct compared to the gold standard. Now that we know how we can measure the performance of TinGa, we will do it on 250 datasets. But we didn't only work on TinGa, we also compared it to 4 other methods. The 3 first methods are the ones that I already showed you in my very first slides. These are the 3 most complex methods that I was talking about. And then slingshot is a method that came out of the comparative study as a very efficient method that is pretty accurate, specifically at identifying simple trajectories. And so here you have the results of all of these methods on a data set that I have already shown you, for which you have the gold standard at the top left. At the top middle, you have the result of TinGa. And you can see that it obtained quite a high mean score. Just for the record, the mean score is saying us how well this trajectory is comparing to this gold standard. But then if you look at race ID, for instance, it did identify this first branch that goes from yellow to green, but then it got quite confused about these purple cells, and so the mean score is quite low. And so you can see the results of BAGA, Slingshot, and Monocle 3. But this is one data set. And we actually did this on 250 data sets. These consisted of 72 real data sets, 178 synthetic data sets of different topologies. Some simple ones, such as linear or cyclic, and some very complex ones, such as connected and disconnected graphs. And here in this violin plot, you can see the results of the different methods applied to the 250 datasets. In the first column, you have the results of TinGa. You can see that it outperformed the other methods on quite an amount of datasets here. You can see that Slingshot also performed very well on some of the datasets. But what is striking here is that race ID got a mean score of 0 for a significant amount of the methods. And this can be explained if you look at this second figure here, which shows the scalability of the methods. On the x-axis, you have the number of cells in the datasets that we used. On the y-axis, you have the time that a method took to run on these datasets. And what you can see is that already for small datasets, race ID was pretty slow compared to the 4 other methods. You can also see that for datasets larger than 5,000 cells, we didn't even retrieve a result for race ID because it asked for too much memory and time to run. The second slowest method was Slingshot, and on the largest datasets that we had, that contained about 12,000 cells, it took about 3 hours to run. On this same dataset, BAGA took about 10 minutes to run, Monocle 3 about 1 minute, and you can see that TinGa was the most scalable of the 5 methods, since it consistently took between 10 and 15 seconds to run. For each of the 250 datasets that we had, we also looked which method outperformed the others. What you can see is that TinGa outperformed the other methods on 88 out of the 250 datasets. The second best outperforming method was Monocle 3, which did best on 60 of these data sets, Slingshot on 52, etc. But I have also told you about the complexity of the data, and we looked at this and color-coded this figure by complexity. The data sets that are represented in the lightest whitish, yellowish color are the most simple ones that contain a linear trajectory. And the data sets that are represented in the dark reddish color are the most complex, in this case, disconnected graphs. And so if we look at the figure closer, we can see that Monocle 3 actually performed very well on more complex data sets, such as trees and graphs. Slingshot, on the other hand, performed best on more simple trajectories, such as linear and bifurcation and cycle. As for PAGA and race ID, STEM ID, they performed best on trees. But if you look at TinGa, it was the method that performed best on both the most simple and the most complex datasets. So in this presentation, I have showed you how TinGa worked with this growing graph that iteratively adapts to the data. We've seen how to see its performance on simple trajectories, and we saw that it performed quite well, but also on complex trajectories. So we have a flexible tool. And we also saw that TinGa was the most scalable of the 5 methods that we used. So next time that you have a new data set to analyze and you start applying a lot of methods, or at least several methods to it, because I hope that we agree that you shouldn't use only one method, but several ones and compare the results. Well, I hope that you'll think of adding TinGa to your toolbox. And I will leave you with the reference to the paper and the GitHub page on which you have an R implementation of the tool. And I thank you for your attention.